What is DNA? DNA is a long, double-stranded molecule that contains the instructions an organism needs to develop, live, and reproduce. It's found in nearly every cell of every living organism, except some viruses, and it's passed from parents to offspring. The full form of DNA is deoxyribonucleic acid. Structure of DNA. DNA has a double helix structure like a twisted ladder. This was famously discovered by James Watson and Francis Crick in 1953 using data from Rosalind Franklin. Components of DNA. Each DNA molecule is made of nucleotides, which are the basic building blocks. Each nucleotide has three parts. First, phosphate group, which gives DNA its acidic nature. Second, deoxyribose sugar, a 5-carbon sugar. Third, nitrogenous base, one of four types: adenine, thymine, guanine, and cytosine. Base pairing: A pairs with T via two hydrogen bonds, G pairs with C via three hydrogen bonds. These pairs form the rungs of the ladder, while the sugar-phosphate backbone forms the sides. Orientation: DNA strands run in opposite directions. This is called anti-parallel orientation. One strand runs 5 feet to 3 feet, the other 3 feet to 5 feet. Where is DNA found? In eukaryotic cells, DNA is mostly in the nucleus and some in mitochondria and chloroplasts. In prokaryotic cells, DNA floats freely in the cytoplasm in a region called the nucleoid. Functions of DNA. DNA is the instruction manual for life. Its key roles include: first one, genetic information storage. DNA stores the code for building proteins. Second one, replication. DNA copies itself before cell division. Third one, protein synthesis. Transcription, DNA to RNA. Translation, RNA to protein. Fourth one, inheritance. DNA is passed from parents to offspring. DNA replication. DNA replication is semi-conservative, meaning each new DNA molecule has one old strand and one new strand. It has three steps. First one, unwinding. Enzyme helicase unzips the double helix. Second one, base pairing. DNA polymerase adds complementary nucleotides. Third one, joining. Ligase seals the fragments on the lagging strand. This ensures genetic continuity during cell division.